Hello, everyone. My name is Seculov, and today I'll be talking to you about a project we're doing in Dr. Kristen Stanford's lab on how signaling lipids regulate muscle metabolism after burn injury. And to give a little bit of context as to why we're doing this research, we know that after severe burn injury, a hypermetabolic state ensues. In this state of heightened metabolism is characterized by catabolic or destructive processes such as increased lipolysis, breakdown of lipids, which is then followed by protein catabolism, the breakdown of proteins, which leads to physiological effects such as muscle wasting. In this muscle atrophy or muscle wasting is of particular interest because it inhibits a patient's return to normal life. This is because the patient undergoes significant loss in skeletal muscle mass. They'll have trouble completing everyday tasks. And as a result, mitigating muscle wasting as well as its effects could improve patient outcomes. And in previous research we've done using rodent models, we saw that after severe burn injury, there was a loss in skeletal muscle mass, but that was preceded by a loss in adipose tissue mass. And specifically in this experiment, we used cohorts of rats, and one group as a control, one group that had their hind limbs burned, and then a third group that had their hind limbs unloaded as well as a burn. And this was to simulate the experience a patient undergoes of being bedridden after severe burn injury. What we saw after that severe burn injury was a loss of adipose tissue mass starting at day 3, and this was followed by a loss in skeletal muscle mass at day 14. Because protein catabolism leads to the loss of muscle mass and lipolysis leads to a loss in adipose tissue mass, we hypothesized that the lipolysis we see after severe burn injury is releasing signaling lipids that then mediate the muscle metabolism response we see after severe burn injury. And to test the differences in signaling lipids after severe burn injury, we used a method of lipidomic analysis or lipid content analysis on plasma samples collected from severe burn injury patients at different time points over the course of 30 days. And in comparison to healthy controls, we found that at these different time points, there were indeed changes in signaling lipid content as well as concentration. And because we wanted to see if these signaling lipids would be having an effect on muscle catabolism, we looked at a particular signaling lipid, an inflammatory marker, 15-HEAT, and its metabolite 15-oxo-E, because they've been indicated in protein catabolism previously. And in comparison to the controls, we saw that in the human plasma data, there were increases in 15-HEAT concentration starting at day 1 after severe burn injury, and this was followed by an increase in 15-oxo-E in days 4 through 12 after severe burn injury. And because this timeline coincided with the loss in skeletal muscle mass we saw in our rodent models previously, we wanted to see what effect 15-oxo-E could be having on protein catabolism. And to test this, we use an experiment utilizing differentiated rodent myocytes or C2-C12 muscle cells. And 4 hours after differentiation, we expose them to various concentrations of 15-oxo-E. And using qPCR gene expression analysis, we saw that there was an increase in the expression of genes related to protein catabolism in atrogen-1, MRF1, and atrogen-5 in those samples that were exposed to 15-oxo-E. And this led us to the conclusion that indeed, a signaling lipid 15-oxo-E could be involved in protein catabolism after severe burn injury. And then because after severe burn injury, a patient undergoes a phase of muscle recovery to recover some of that muscle mass they lost originally after their injury, we wanted to see if another signaling lipid could be involved in this response, in the muscle recovery response. And we looked to an anti-inflammatory marker, RVD1, resolvin-1, because it's been indicated to be involved in muscle recovery and regeneration, and it's secreted from adipose tissue. And we saw that resolvin-1 was increased in days 7 through 25 after severe burn injury in those human plasma samples. And because this coincided after increase in 15-oxoate concentration, we wanted to see if RVD1 could be having a response in rescuing that protein catabolism we see after severe burn injury. And to test this, again, in another experiment, we used the C2C12 muscle cells and exposed them to concentration of 15-oxoate, as well as RVD1 and 15-oxoate concurrently. And what we saw was that, as expected in comparison to the controls, the myocytes exposed only to 15-oxoate had an uptick in their protein catabolism genes. And then these genes were then downregulated when in groups exposed to RVD1 and 15-oxoate. So this led us to the conclusion that RVD1 could be mediating a protein anabolism response we see after severe burn injury. And to go over all we've found so far in this project, we saw significant changes in signaling lipids in human samples after severe burn injury. And one of these signaling lipids, 15-oxoate, could potentially be involved in protein catabolism. And another signaling lipid, RVD1, could be involved in the protein anabolism response we see after severe burn injury. And some future directions this project could go include evaluating if we see similar results in muscle tissue as well as muscle cells, and therefore to evaluate translational viability into human samples. And with research like this, as well as other investigation, we hope that this will contribute to the development of therapies that help mitigate muscle atrophy after severe burn injury, and then aid patients in returning to their everyday life. And with that, I would like to thank you for listening, and please leave your comments below. Thank you.